Students, today we will see from anatomy. All right, we will solve some question, and along with the question we will discuss. So our first question is, which is enucleated at maturity? The cell without nucleus. Okay, here our options are sieve cells, companion cells, cortical cells, and palisade cells. Okay, out of these, the sieve cells, okay, this is without nucleus. The sieve cells, this is component of phloem tissue. The sieve component is when it is young, they have nucleus, but when it is mature, nucleus is degenerated. So at maturity, though the cell is a living cell, this is a living cell without a nucleus. Okay. Here is, I think you can see this is the structure, anatomical structure of the phloem, and here is the sieve cell. This one is the sieve cell. These are the companion cell. Okay, companion cells, they have prominent nucleus, but the sieve cells which make the sieve tube, they don't have nucleus when they are mature. This sieve phloem parenchyma also, they have this prominent nucleus. So it is the sieve cells of phloem, they don't have nucleus. Of course, the other tissues, in other tissue like Sclerenchyma, okay, then xylem vessels, um, these they also don't have nucleus. They are, of course, they are dead cells. All right, they are dead, so naturally they don't have cytoplasm and nucleus both. But the sieve cell is living and don't have any nucleus at maturity. Next question is, lignified cell wall occurs in, out of this, the lignified cell wall. Lignin is a cell wall component which is present in some of the type of cells. So those cells are known as lignified cells, and here the lignified cells are the xylem cells. This is cambial cell is number D here. This lignified cell wall in the xylem cells. In the xylem cells particularly, this say Sclerenchyma, tracheids, and vessels. These are the component of the xylem. They are lignified. Sclerenchyma tissue, that sclerenchyma also is lignified. So lignified cells in this option is xylem cells. Of course, here xylem cells, in the xylem, some xylem parenchyma is found. That xylem parenchyma is a living cell. Okay, xylem parenchyma is living and it is without lignification. Next is monocot root differ from dicot root in having a monocot root. Monocot root differ from dicot root in having dicot root and monocot root differ in well-developed pith. They have well-developed pith. Pith is well-developed. Here you can see it. We have diagram for is the diagram of dicot and monocot root. This is here is the upper one. This one is dicot root and this one is monocot root. Of course, in this diagram, those things are not very clear. Here also, this is the pith or medulla. Here also is pith is there. In this diagram, they have not given proper diagram in the book. This is one way it is wrong. That in dicot root, pith is either small or absent totally, and in monocot the pith is prominent. It is large. Okay. So, and another difference of monocot and dicot is here you can see the number of xylem, bundles. Okay, here it is 4, but in this case number of xylem bundles are many, 1 2 3 4 5 6 7 8. Okay, nearly 8. So here in monocot, xylem bundles, actually we have to say xylem bundles many. Here it is less in number in monocot. Here usually 2 to 6. Okay, 2 to 6, very common is up to 4, but in case of monocot it is more than this. Next question. Yes, do you have any problem? Can you hear me? Hello.
Yes, sir, we can hear you.
Do you have any query, any problem?
No, sir.
Okay. So here it is. Our answer is that well-developed pith is there. Well-developed pith is the answer. Next question. In this, healing of wounds, there is a little spelling mistake. ND. Okay. Healing of wounds occur due to the activity of which is due to the activity of due to the activity of, it is secondary meristem, primary meristem, apical meristem, intercalary meristem. Say anywhere there is, if there is any wound, so that healing is done by the production of new cells. That is addition of new cells, and this addition of new cell is always with the meristem, because meristems are the tissue which divide continuously and produce new cell. But which meristem? Here actually it is the secondary meristem. right? This one is secondary meristem, not by the primary meristem. Primary meristem is that primary meristem developed from promeristem. Okay, and promeristem is the one which is present from the embryo. That is when the plant is organism in the embryonic stage. From that time, the embryo meristem present, that is known as promeristem, and promeristem give rise to the primary meristem. But the secondary meristem, this one developed from permanent tissue. Okay, that the permanent tissue, permanent cells which are there as surrounding, say if there is one wound here, injury here, then the cells surrounding it, they will change into meristem by the process we call de-differentiation. Okay, by de-differentiation it change into meristem. So as this meristem is from the permanent tissue, so this is known as secondary meristem, and this secondary meristem heals that wound. And once the wound is healed, then the meristematic activity of the secondary meristem is again change. So that meristem again change into permanent tissue. That is by call it is redifferentiation. So there are terms differentiation, de-differentiation, and redifferentiation. Okay. Apical meristem, of course, it is primary meristem. Intercalary meristem also is primary meristem. The another secondary meristem is that cambium, particularly Cork cambium. Cork cambium is secondary meristem. I think you should remember all this. And the vascular cambium is mixed. It is primary as well as secondary meristem. Come to the next question. This next question is conjoint, collateral, and closed vascular bundles are found in conjoint, collateral, and closed. The dicot and monocot. This is particularly in case of a stem, dicot stem and monocot stem. In the dicot stem, vascular bundles are conjoint, collateral, and open. Okay, and this one in monocot, it is closed. In monocot, it is closed. Open and close here, actually there is, in case of this, within the vascular bundle, there is cambium. Cambium present. That's why it is called the open vascular bundle, and here the cambium is absent. So the further growth or secondary growth, particularly, it is closed. There is no further growth takes place, but as in the dicot plant, within the vascular bundle there is cambium tissue, so further development, further growth takes place. So this is open for growth. So here we have, in case the grassroot, grass stem, sunflower stem, cucurbita stem. Say this sunflower stem, okay, and the cucurbita stem, they are dicot. So in these plant we don't get close. Here the vascular bundles are open, but in the grass stem the vascular bundles are closed. Root, in the root the vascular bundles are neither closed nor open, because vascular bundles are not conjoined. In the root the vascular bundles are radial. Radial means here the xylem and phloem, they are found in separate patch and alternate each other. Okay, xylem phloem alternate each other, and that's why it is called radial vascular bundle. Radial vascular bundle is the characteristic feature of monocot or characteristic feature of root, and in stem it is conjoined collateral. Heartwood or duramen is heartwood or duramen. There are terms for that. Heartwood, and another term is called sapwood. Okay, heartwood and sapwood. This is also known as duramen, and this one is alburnum. Okay, so this thing, heartwood and sapwood, these are formed after the secondary growth in the stem and root. This is, here the answer is, options are heartwood or duramen is inner region of the secondary phloem, outer region of the secondary phloem, inner region of the secondary xylem. This one is the answer. Inner region of the secondary xylem. The wood, term wood is always used for xylem tissue. Xylem is equivalent to wood. Okay, so it is found in the inner region of this. Say here, sorry for there was some technical problem. Okay, so this is the But so heartwood and the sapwood, there is actually during the secondary growth, okay, during secondary growth when it takes place, this tissues that is formed towards the center, the xylem tissue which has been secondary xylem tissue which has been formed in the earliest stage, all right, earliest stage, those tissues become dark and gradually dead. So they become dark and different chemicals etc. are secreted. So this is usually dark color and very hard. They have thick cell wall. So this portion of the tissue is known as heart tissue. Because this is at the center, towards the center, heart, heartwood. All right. And those xylem tissue which has been recently formed, all right, they are usually slightly light in color and they are known as sapwood. Sapwood is, this is a lighter, light color. Okay. And this one is physiologically functional. Functional, that is conduction of water etc. takes place through the xylem of the sapwood, and heartwood cannot carry out this transportation of water. So heartwood only provide mechanical support. This is next question is youngest secondary xylem occurs just inside the cork cambium, just outside the vascular cambium, or just inside vascular cambium, just inside the secondary cortex. So what is the position of the youngest secondary xylem? Say in the when secondary growth takes place, say if this is the cambium ring, this red portion is the, say it is the cambium ring, all right, this is xylem recently formed, and outside this side is phloem, this is secondary phloem, this is secondary xylem. So it is new xylem tissues, youngest xylem, secondary xylem tissue, they will be near to the near to the vascular cambium, and those which are away from the vascular cambium, they are older and older, and very near to the secondary, very near to the cambium are the younger one. So towards the cambium it is younger, and away from the cambium it is older. So youngest secondary xylem will occur just inside the vascular cambium. just inside the vascular cambium. Like that also on the outside, as it is phloem, so younger secondary phloem will be near to the cambium ring and older one will be away from the cambium ring. This is all the primary xylem, primary phloem. It will be primary xylem will be near the center. Okay. Next one, age of the tree is calculated by height, girth, number of branches, and number of annual rings. Number of annual rings. Annual ring, or it is also known as growth ring. During secondary growth, there is formation of what we call that springwood and autumn wood. Okay, springwood is also known as early wood and late wood. This autumn wood is late wood. This actually what happened that during the early stage of the growth or spring, spring season is the most favorable season for the growth and development. So the secondary growth or the tissues, secondary tissues which are produced during the secondary growth, they have large vessels, large vessel cells. Okay, large vessel cells, so they have big lumen. Lumen means cell cavity, and the quantity also is more in quantity. Okay, and slightly they are light in color. But when the unfavorable time come, that is the autumn, winter, okay, this one is unfavorable. So during this period, the secondary growth is very less secondary growth. All right, and cells are cells, small cells, and quantity less. This one, due to these smaller cells, unfavorable condition, quantity is less. So this is, they are slightly dark in color. Dark color, here it is light colored and large quantity. So what we can see in a tree or in its trunk, we can find repeatedly dark light bands. Okay, dark light and dark light bands are due to the formation of springwood and autumn wood. Again springwood, again autumn wood. So in this, in a year, one springwood and one ring of autumn wood is formed, and these two together, as it is produced annually, one in 1 year. So this one is known as annual ring, and one annual ring is formed in 1 year. So the age of the tree can be calculated, determined by counting the number of annual rings. Okay, so this is the by way that. Now come to this question. This is one that match the following and choose the correct option. So here this side is meristem, parenchyma, collenchyma, sclerenchyma, epidermal tissue. All right, and this side it is photosynthesis and storage, its function or other properties, mechanical support, actively dividing cells, stomata and sclerid. Say what will be the matching for this? Meristem is actively dividing cell. Parenchyma, that is, parenchyma make what? Parenchyma, they can carry out photosynthesis and storage. The parenchyma cells with chlorophyll, called sclerenchyma. This sclerenchyma tissue carry out photosynthesis, and they also store food, that is, produce food. So it is collenchyma is collenchyma. Let us leave it for time being. Sclerenchyma. Actually, for sclerenchyma tissue, they can give mechanical support. See here, sclerenchyma is a very hard tissue and can give mechanical support. Mechanical support. And sclerenchyma, here is another one, that sclerids. Sclerid is a type of sclerenchyma tissue. Type of sclerenchyma. Because sclerenchyma is two type. This one is of two type. One is fiber and another one is sclerid. All right, fibers are long, thin, but sclerids are short and of any irregular shape, of any shape. So this one, this sclerenchyma can match with the mechanical, with the sclerids. But epidermal tissue can form the photostomata. All right, you know that stomata are on the surface of the leaf surface. So this stomata, those which are made of guard cell, these guard cells are modified epidermal cell actually. Guard cells are epidermal cell. So this one will match with this. Now here you should have confusion that mechanical or sclerid. So the sclerid Sclerenchyma cannot fit with sclerid. Sclerenchyma is not sclerid. It is, so here we will match Sclerenchyma with this sclerid, and Sclerenchyma we will match with the mechanical tissue. Actually, in case of monocot, where this Sclerenchyma tissue is found in the hypodermis, they provide mechanical support because they have irregularly thickened wall and no intercellular space. So they can provide. Now, then what is 1 is with 3? Which one is 1 with 3? So only this one is going with meristem, actively dividing cell. Actually, if you see, this option to determine is very easy, because if you know that meristems are actively divided cell and here A is not having any other match, only option B is showing that this one is with actively dividing cell. So ultimately this will be the answer, not this, not this, not this, because A, meristem is not matching with sclerid, meristem is not matching with photosynthesis or not matching with mechanical tissue. Okay, so our answer is option B. So if you have that idea of some, at least one or two of these, then you can match these things. Next, we will go for another matching. Okay, this one we have already done. This is also the same one. Similar way, we will do another matching question. Match the following and choose the correct option from below. Here, for this side is cuticle, bulliform cells, stomata, and epidermis. And this side in the column B or column 2, and this is column 1. Column 1, this side is guard cell, single layer, waxy layer, empty colorless cell. So what will be the matching? Can it match? See here, cuticle present. This is cuticle is made of a chemical known as cutin. Okay, so and this is usually formed on the outer surface of all the aerial parts of the plant, on the leaf surface, on the stem surface, on fruit surface, on flower surface, that the epidermis is there. Epidermis is covered by, there is a layer on the epidermis, this is the cutin layer, and this is actually, this cutin is the waxy layer. So cuticle will match with this. Bulliform cells, this is bulliform cells. This is also known as motor cell. Okay, these cells are found in grasses, grass leaves, and some other plants. Grass leaf epidermis. These cells in the epidermis, these cells are larger in size than its neighboring cells. Okay, and these are little bit of hollowness is there. This cell can absorb water, can release water. When it absorbs water, then this will help the leaf open. Okay, It is a leaf is stretched when the bulliform cells are having enough moisture. So they increase in volume and this leaf is open. But when there is a drought condition, dry condition in the within the plant, there is very less water, or the transpiration has taken place very fast, so the leaves has lost water. In that condition, when there is a dryness is there, this bulliform cell will shrink, and as they will shrink, then the leaf will curl. Okay, it will curl and leaf curling takes place, and by this way it reduce the exposed surface and transpiration is less. Sun is reduced. So this is one way, one of the adaptation for the dry condition. So sometime I think in the dry season you can see in some of the plant in grasses that the leaves has been curled. Okay, it has become narrow. So bulliform cell, these are the empty colorless cells. They are large empty colorless cell. A stomata cell will match with the guard cell. Okay, epidermis is usually the single layered, double layered epidermis, of course, found in some of the plant uh, um, in some of the xerophytic plant. Okay, so usually the epidermis is single layer outermost. So with which it will match? Here also see A will match with 3. Say, in this case, in this option, there is a confusion. By knowing this first one, you cannot decide that your answer will be this, because in all the 3, this 3A is matching with this 3. So you cannot, you have to see the next one. B is bulliform cell, B with the 4, empty colorless cell. So B4 is in this one. So naturally you decide 2 are correct. So means this will be this one. C is 1. Of course it is 1. D is 2, single layer. So by deciding, by knowing 2, you can go for this answer. Not necessary to know that all the, if 1 or 2 you don't know also, you can make the answer. But one thing you remember, that sometime what happened, that these numbers, okay, there's a column in the 2 column, the numbers of option they may be more. So sometime what happened, okay, this side there are 4 things, but this side in column 2 it may be more than 4, it may be 5, 6. In that case this matching is become difficult. Then come to phallogen and phylem. Okay, these two has been typed together. Phylem. Okay, phallogen and phylem respectively denote cork and cork cambium, cork, secondary cortex, cork, and the secondary growth. All right. This secondary growth in the stellar region it is by vascular cambium. But in the extrastellar region, outside the vascular area, there is in the extrastellar region means outside this vascular cylinder. Here, the secondary growth takes place by cork cambium. And this cork cambium produce two things. Say, cork cambium produce two things. On the outside, it produces cork outer, that is on the surface of the stem, and on the inner side, it forms the secondary cortex. Okay, secondary cortex. This cork cambium has another name. This is known as phyllogen, and the cork is known as phylem. And the secondary cortex is phylloderm. All right, this is the alternative name. Here is say this is a diagram of diagram of a portion. Here you can see this portion. You can see here this portion. Now this is the cork cambium, right? And on the outer side, it is producing a nicely arranged cell. This is the cork, and this one is cork is called, that is phylem. And this side, on the inner side, this tissue, this portion of the tissue, this is the secondary cortex, or it is called phylloderm. Okay, this one is phylem. So, and this cracked structure loosely, you can see, this is known as lenticels, or lenticels. These lenticels become very important for the exchange of gases, because here the cells are very much loose cells. So exchange of gases are there. So now our answer will be phyllogen, phyllogen is phyllogen and phylem. So phyllogen is that is the Cork Cambium, phelogen is Cork Cambium, and phloem is the cork. Phylodermis, secondary cortex. Here you can see the secondary growth, that is, this is the vascular cambium. This side is xylem formation, this side is secondary phloem. Secondary phloem, this side is xylem. Next one, which of the following is made up of dead cells? Dead cells, which one? This is in NEET 2017, it has come. This is dead cells. Dead cells are here. Phloem, Sclerenchyma, phloem, xylem, parenchyma. Say this xylem parenchyma is a living cell. Xylem is a dead tissue because xylem vessels and tracheids are dead. Phloem is mostly it is a living. Sclerenchyma is a living. This is all living. This one is a living. This is mostly living. Okay. Phloem, here in the diagram we have got that phloem means cork. Cork is made of dead tissues. Here their cytoplasm, when the cork a little bit mature, their cytoplasm is dried and they have the suberin layer. Suberin or wax layer is found on the surface. So this layer, this cork layer become impervious to water. So it protects from the water loss. Next one is specialized epidermal cells surrounding the guard cells are called what? Say this is the guard cells. Okay, here you can see the diagram, very big. This is the guard cell. This one is a normal dicot plant, and this is the guard cell of monocot grasses. Okay, here in the dicot, the guard cell is kidney shaped or reniform. Okay, but here it is dumbbell like. See, two balls on two ends, and in the middle one is thinner. So this is the in guard cell in the in case of grasses. All right, here is, this is guard cell. Say here, this one is guard cell. All right, this chloroplast are there because guard cell carry out chloroplast, and these cells, some of the cells surrounding the guard cell, their number may be 2 or more. So 1, 2, 3, 4 are here. These are also epidermal cell, but these are slightly special, all right, other than the other rest of the epidermal cell, and these cells are known as subsidiary cells. Okay, so it is the, our answer will be subsidiary cells. Okay, today we will stop here, all right, because it is time also, and there are of course many questions. You can solve it. If there is any problem, you can ask, right, or you can send the question through your coaching center. Okay, thank you. So we will close it. Now, do you have any question? Any query? Hello? No.